Metaphase reminds me of the fact that they line up in the middle.
So these little chromosomes line up in the middle of the cell. The nucleus is like gone. You can see their little belt still. Hasn't been broken yet. Indiana Jones is on either side. So chromosomes line up in the middle. Indiana Jones, who's purple, gets out his weapon of choice. It's a whip. And he flicks the whip onto these little belts to rip this apart. Here comes that whip. Indy attaches his whip to the belt. These little things. I am now going to decode this into normal human language. Indy is actually a centriole. Attaches. The whip is actually called a spindle fiber to the belt. And the belt is not called a belt. It's actually called a centromere. Centromere. So you could say this in regular language. Indiana Jones takes out his whip and snaps it onto the belt. Or in science, the centriole attaches the spindle fiber to the centromere. Here's how I feel about this one. Makes sense? Science language. Done with metaphase. What do you have to know? Chromosomes line up in the middle. Indiana Jones gets out his whip. Now we're going to move on to Anna the Ripper. Like Jack the Ripper, we're going to rip those chromosomes apart. Remember, we want a copy to go into one cell and a copy to go into another cell. So this side will go into one cell. This side will go into the other cell. One of them will go here. One of them will go here. We're going to pull these apart. So if you look at them, that little belt snaps.
Because Indy broke it with his whip and pulls the two pieces apart.
For a moment in time, this is a chromosome. These pieces, for just anaphase, are called chromatids. Chromatids, another chroma word, moves to opposite sides of the cell. We are yanking this cell apart to put it into two cells. Telophase! Daddy, the telephone is ringing. We have taken these and we stick them in a new nucleus.
Well, Indiana Jones is done. Puts his whip away.
Two new nuclei form around the separated, organized chromosomes that we've doubled and now ripped apart, one set for each cell. We're going to cut it right here. So all you need to know for telophase
is that two new nuclei form at telophase.
And now we're just going to get some scissors and we're going to cut this cell in half. This cell's very happy. It's going to have 46 total. This cell's very happy. It's going to have 46 total. We've now cloned our cells. These are called daughter cells. Sorry, sons. They're called daughter cells. They are identical. They are smaller. And they are going to go right back into interphase where they go do normal cell stuff. Once they go back into interphase, they will go do normal cell stuff. They grow here and they'll start this whole process over. Your cell spends most of its time here growing, keeping you alive, doing whatever its job is, like helping you see or being a heart cell and pumping, whatever it's doing. Plus, it's always copying your DNA. That's a lot of work because it's 3 billion bases it has to copy. And of course, it can't make a mistake. We're going to stop there and we're going to move on to a lab online where we look at some of these cells in each phase.